Hello, this is the presentation for the Rapid COVID-19 Assay Manufacturing where we, Michael, Zeus, and Rachel, with the help of Dr. Christy Peebles, modeled the manufacturing scheme for creating a rapid paper-based test for COVID-19. This presentation will cover the scope, motivation, problem, competitive and market analysis, process synthesis thus far, our key assumptions, current insights and findings, and future steps for us and future groups working on this project.
Hi, I'm Rachel, and I will be presenting on the background and scope of this project. The background for this project is the COVID-19 pandemic that has been impacting lives for over a year now. Rapid screening and limiting the spread of the virus have been critical in prevention and control. An important part of the background of this project is also improving the access of COVID-19 tests for those in the developing world. This project proposes a method to make a paper-based assay that tests for viral RNA in an easy and efficient way. Other tests like this have been developed for the Zika virus. A paper disc holds the freeze-dried cellular machinery to induce a colorimetric change and indicate if viral RNA is present in the tested sample. The paper-based assay uses an RNA toehold design and a cell-free system to perform this operation, but more on that later. The motivation of this project stems from the global pandemic of COVID-19. While some tests have been available to us at home, some places are not so lucky and don't have the ability to test as widely as we have. This test would be a fast and inexpensive way to test for the COVID-19 virus. Its freeze-dried state would allow for it to be shipped worldwide and used in places without access to laboratory settings. This easy test could also allow for the testing of individuals who may be asymptomatic carriers, further preventing the spread of COVID-19. Beyond that, this test could help to optimize treatment for individuals who are caught early by this test. Overall, this test is easy to use because it bypasses much of the laboratory techniques that must be performed for other widely available tests. The goal associated with this project is to make a manufacturing model which will yield 1 million of these paper-based tests. Only a few microliters are necessary to freeze dry onto paper discs. So this goal is achievable in seeing if manufacturing this many tests is something that can be done. For this test to function, it must have multiple components that are freeze-dried onto the paper disc. The first of these components is an RNA toehold switch that arises from a plasmid within transformed E. coli. An RNA toehold switch works by having a piece of RNA that is held in the loop by complementary base pairs. Following the loop in the direction of translation is a gene that, when translated, leads to a protein that gives a colorimetric change. However, this part of the process only works when there is a trigger RNA, which, in this case, would be the viral RNA contained in COVID-19 viruses. The translation step occurs on this paper test by the presence of cellular components that come from broken up E. coli.
The market analysis for this product is rather unique given the circumstances. An unprecedented scenario of nearly unlimited demand for testing existed at the height of the pandemic. Since then, the number of tests administered daily in the US has been slowly but steadily declining over the spring months. This trend is visualized in this figure, where the bars represent the total number of tests administered each day in Colorado. However, there are still millions of tests being performed in the US each week, and this seems likely to continue for at least several months. As COVID restrictions are lifted and crowds begin to gather again, the need for screening tests will be as relevant as ever. A cell-free paper-based test is fast, easy to use, and ideal for screening purposes. This assay could help bridge the gap from lockdown back to normalcy. This cell-free assay technology can also quickly be adapted to detect other viruses besides COVID-19, ensuring future usefulness. For a competitive analysis, we researched different kinds of COVID-19 tests currently in use. There are two main kinds of tests, each filling a niche in the market. The first kind are molecular tests. Molecular tests can take days to produce results. The tests often need to be administered by a professional. Samples need to be refrigerated and transported, and obtaining results requires complex lab equipment. However, these tests are the most accurate, and these characteristics make molecular tests ideal for making or confirming a diagnosis, but not for screening purposes. A more direct competitor to a cell-free paper-based assay would be an antigen test. Antigen tests can produce results much faster than molecular tests, though the results are less accurate. Antigen tests are ideal for screening purposes and fill a similar market niche to a cell-free paper-based test. The advantages of a cell-free paper-based test include rapid results, ease of use, and a minimal equipment requirement. Testing can also be done on-site with no need for refrigeration or a lab. The cell-free assay could provide better testing in remote and developing regions than what is currently available.
In our preliminary process synthesis, multiple different options for the manufacturing of this rapid COVID-19 test were considered. The first considerations were about how a cell-free protein system was going to be developed and how the plasmid for the toehold switch was going to be acquired. For economical and convenience reasons, a transformed E. coli containing the plasmid to make the toehold switch would serve to also provide cellular components necessary for a functional cell-free protein system. A batch reactor following a seed train for growing of the cells was chosen because of the wide use of batch bioreactors for applications such as this. The cell disruption step frees the cellular machinery for use in a cell-free protein system and yields cellular components which can be repurposed for future protein synthesis in the colorimetric portion of this assay. A separation step is utilized to remove excess liquid and concentrate the cell-free system components and toehold switch components. The addition of buffers allows for the cell-free system to be viable later with necessary building blocks like free amino acids and nucleotides. Lyophilization or freeze drying is the step where the cell-free system, complete with RNA toehold switch components, is added to the paper disc. After freeze drying these components onto the paper disc, packaging and distribution can occur where this test can be kept at room temperature until it will be rehydrated and used with a sample. This is the process synthesis for a paper-based rapid COVID-19 test as modeled in SuperPro Designer. First, the transformed E. coli cells are grown in a 300 milliliter inoculation system and are given nutrients such as glucose while they are incubated at 37 degrees Celsius. Then, a seed train process is used to grow the media so that the next reactor is 10 times the volume of the previous reactor. This is done until there's 300 liters of media available for the bacteria to grow in. To utilize the cell-free system, a French press is used to break open the cells by forcing the cells through a very small opening, which disrupts the cell membrane due to pressure differences. Next, a centrifuge is used to separate the solids, such as biomass and cell machinery, and liquids, such as water and aqueous glucose. A buffer is then used to help with the function of cell machinery later, and then the solution is dried using freeze drying for preservation and distribution. Some key assumptions made in the making of this model are that some procedures as listed here have ideal conditions. The fermenters were assumed to be perfectly mixed such that there are no concentration differences throughout the reactor and such that the cells are not destroyed by the mixing. It is also assumed that 3 microliters of the final product will allow for this system to be functional for the detection of COVID-19.
Some insights and findings of this project include the following. The assay development group achieved a visible color change in the COVID-19 assay in progress, showing promising potential for the effectiveness of the assay. A test for Zika virus on a paper disc has already been developed and functions in the same way. The team developing this test in the lab found that this COVID-19 test is even faster than the Zika assay. The development group also found that the paper disc can function with just 3 microliters of product, This allows for the manufacturing goal of 1 million tests to be easily achieved with the 300-liter fermenter in the model. This model for manufacturing the assay is now developed and can be modified in the future for different applications or to incorporate future research. It was also determined that the unique qualities of a rapid cell-free COVID-19 assay could provide advantages over tests currently in use, such as requiring little technical skill to administer the test.
In conclusion, it was learned that this assay can be applied to other viruses with simple modification of the plasmid carrying the RNA toehold switch. The goal of 1 million tests using a 300-liter bioreactor is achievable with a concentration of 50 grams per liter of the final product. It was found from our idealized model 57.32 million tests could be made. This larger number is possibly achievable, but our goal of 1 million tests is a realizable amount from our model. If more tests can be made from this model, then that serves just to lower the estimated cost of production of this rapid COVID-19 assay. Next steps for this process include fine-tuning the process synthesis, evaluating the cost of production and sustainability aspects, investigating the bottlenecks further, eliminating roadblocks that may occur, performing more laboratory tests for the actual manufacturing process, scaling up the process to a facility-scale production, and evaluating the safety of this manufacturing process.